We're going to take another look at epigenetics and learn a bit more about how it's a study that is very relevant to our everyday life. Except for mature red blood cells and cornified cells like hair and nails, which do not have a nucleus, every cell in our body contains the same set of DNA we inherited from our parents. If every cell contains identical genomes, how do we have so many different populations and types of cells that are assigned to carry out different functions? The answer to that is that not every gene is turned on in every cell, and proteins produced by cells within a population can also vary depending on cell types. We call this differential gene expression. Our cells accomplish this at different levels of regulation. Differential gene transcription, selective processing and translation of RNAs, and differential protein modification. We will focus on the first level of gene regulation, which happens pre-transcriptionally. You have probably heard about cells receiving external or internal signals, like growth factors, or the nutrient status that triggers or represses transcription of particular genes. But did you know that there are changes that can be made to a DNA molecule or its histones that can affect whether a gene is turned on or off? We're not talking about mutations which are permanent alterations to the DNA sequence. No, these changes are reversible and can happen during your lifetime. Some of these changes can even be passed on to your children and their children. We're talking about epigenetics. There are three main epigenetic mechanisms: methylation in a DNA sequence, post-transcriptional modification of histone tails such as methylation, acetylation, and phosphorylation resulting in chromatin remodeling, and RNA-based mechanisms. In your other lectures on epigenetics, you will have seen the effects of histone modifications. So, in this presentation, we will focus on DNA methylation. We will also look at how exposures to external factors, such as nutrition, can play a crucial role in influencing gene expression through DNA methylation and demethylation. Both eukaryotic and prokaryotic DNA can undergo methylation. While we have always known that in bacteria, both cytosine and adenine can be methylated, scientists thought that in eukaryotes, methylation only occurred on cytosine. Recently, it has been discovered that adenine does indeed get methylated in most embryonic stem cells. But for the most part, methylation happens on cytosine in eukaryotes. Cytosine can be methylated through conversion into 5-methylcytosine by DNA methyltransferase enzymes, or DNMT. The majority of methylation occurs in CG dinucleotide context. Regardless of whether methylation takes place within or outside the CG context, cytosine methylation is crucial to genomic imprinting, X inactivation, gene silencing, and repression of transposable elements. If the methylation process is altered in some way, gene expression can become dysregulated, and this can contribute to serious diseases like cancer, autoimmune diseases, metabolic and neurological disorders. The human genome contains approximately 30,000 CpG islands. The P stands for phosphate in the cytosine phosphate guanine motif. CpG islands are clusters of C-gene dinucleotides in which cytosine occurs immediately 5' to guanine, and they form stretches of DNA about 300 to 3,000 base pairs long. This image shows the typical distribution of methylated versus unmethylated CG islands. Note that most of the CG islands found in the promoter region and near the transcription start site are mostly unmethylated, while the ones within the gene body and transposable elements are methylated. In fact, more than half of human genes initiate transcription from CpG islands. These include housekeeping genes that are expressed in all cell types, as well as a large number of master developmental regulators like the Hox genes. Recall that housekeeping genes are genes required for basic survival of a cell, so they are found in all cell types. Recent studies show that CpG islands play a role in initiating and regulating transcription, and CpG islands residing within genes function as alternative promoters. What we see is that methylation found in promoter-associated CpG islands is generally correlated with low or no gene expression. Alternatively, if the gene happens to be a repressor and its promoter region becomes methylated, expression of the repressor is usually inhibited, and previously repressed activities, like transcription of certain genes, are enabled. So, since methylation is usually associated with turning off gene expression, it is generally accepted that methylation of intra and intragenic CpG islands represses their activity as alternative transcriptional promoters. What we also find is that while different tissue types present with significantly different methylation levels, as mentioned before, CpG islands associated with promoter regions are found to be almost always unmethylated. In fact, only less than about 3% are methylated, whereas many of the ones located inside gene bodies are methylated. In this figure, you can see that hypermethylation generally occurs within gene bodies, which represses alternative transcription start sites, while promoter regions are hypomethylated, which leads to gene expression. Repeats are also hypomethylated. What is interesting is that in certain contexts, genes with unmethylated CpG islands in the promoters appear to be suppressed, while promoter hypermethylation has been reported to upregulate gene expression. The location of the methylated CpG islands from transcription start site appears to influence transcription regulation. It was also found that some transcription factors prefer to bind methylated rather than unmethylated CpGs. Due to the presence of enhancers in the first intron, which are bound by transcription factors, methylation of the first intron is also associated with gene expression in certain genes and cell types. Although there's still a lot about epigenetic mechanisms that we don't fully understand, we do know that DNA methylation is one of the key mediators of gene expression, and they also keep repeat sequences and transposable elements in check. Aberrations in the methylation machinery can contribute to a variety of disease processes. Here are a few examples that highlight the link between epigenetic changes and diseases. Genome-wide hypomethylation is considered a hallmark of cancer. In particular, repeat DNA is often observed to be hypomethylated in cancer. Mutations in DNA methyltransferase 3A, as well as the methylcytosine dioxygenase enzyme TET2, which promotes DNA demethylation, are commonly found in patients with myeloid malignancies like leukemia and T-cell lymphoma. An example of altered histone modification leading to cancer is mutations involving the methyltransferases of histone 3 lysine residue 36 or H3K36, which are frequently seen with hematological and solid tumors. For example, multiple myeloma, pediatric acute lymphoblastic leukemia, and renal cell carcinoma. These mutations are associated with poorer outcomes and relapses. Two examples of common diseases associated with epigenetic changes are rheumatoid arthritis and multiple sclerosis. Rheumatoid arthritis is a chronic autoimmune disease characterized by joint inflammation with eventual destruction of the cartilage and bones of the affected joints. In some patients, multiple body systems are also affected. A genome-wide DNA methylation analysis showed that altered DNA methylation of human leukocyte antigen class II, or HLA2, is linked to the risk of developing RA. In early RA, the level of DNA methylation at various sites in the hedgehog signaling pathway is associated with the patient's response to treatment. Recent data also suggest that DNA methylation mediates the risk of developing RA due to smoking in people with certain genotypes. Multiple sclerosis, or MS, is another chronic autoimmune disease that's also neurodegenerative. Changes in DNA methylation of multiple immune cell types and in patients' hippocampi have been observed. Smoking, which is a risk factor for MS, can change the DNA methylation in MS patients with certain HLA haplotypes. Other examples of health-related phenotypes associated with epigenetic changes include hyperglycemia, obesity, schizophrenia, and Parkinson's disease. So, we've seen a little bit about how external influences like smoking in conjunction with genetics are associated with disease processes. Now, let's take a very brief look at another major environmental exposure that can affect our epigenome. Like the name suggests, nutritional epigenetics examines the interaction between food and epigenetic effectors, as well as the resulting alterations to patterns of gene regulation. Nutrient levels and bioactive dietary components can make epigenetic changes that are potentially reversible, but they can also be inherited through multiple generations. Nutrients can directly inhibit epigenetic enzymes like DNMT or alter the availability of substrates needed for the reaction to occur. Folate is a water-soluble B vitamin and the carbon source required in the synthesis of S-adenosyl L-methionine, otherwise known as ADAMET or SAM. ADAMET is a methyl donor of numerous important methylation reactions. This is just showing you how folate donates a carbon here to make S-adenosyl methionine. Please do not memorize this. There's no need for you to know the different components and details about the pathway. An increased concentration of ADAMET can protect the genome from global hypomethylation by stimulating DNA methyltransferase reactions. We get folate from leafy green vegetables like spinach, peas, kale, and even asparagus. Folic acid, which is a synthetic form of folate, has been added to our food supply in things like bread and cereal since 1998 in the US and Canada to prevent neural tube defects such as spinal bifida in newborns. Several studies have found that a higher dietary intake of folate is associated with a reduced risk of type 2 diabetes. Type 2 diabetes patients were found to have lower levels of folate in circulation than control subjects. Biopsies taken of their liver also showed a significantly decreased level of DNA methylation compared to the control subjects. In an in vivo study in mice, the gene expression of P16 tumor suppressor is correlated with dietary folate intake. The silencing of this gene's promoter is frequently seen in breast cancer. Other cancers associated with hypomethylation due to low folate intake are colorectal and pancreatic cancer. Choline is another methyl donor nutrient which can alter DNA methylation status and therefore gene expression. Choline is needed here to make betaine, which maintains this methionine homocysteine cycle and enables the synthesis of S-adenosyl methionine. Again, please do not memorize this pathway. We just want you to know the main concepts, and I'm just providing this information as context so that you know why choline is important to the epigenetic processes. Choline-rich food includes beef liver, eggs, fish, nuts, cauliflower, and broccoli. A number of studies have found that the Mediterranean diet, which is higher in plant-based and choline-rich foods, is associated with reduced risk of heart disease and a reduced incidence of cancers. Another study demonstrated that mice fed a high-fat and high-sugar diet prior to and during pregnancy results in epigenetic changes in the offspring's renin-angiotensin system, or RAS, which led to altered gene expression of angiotensin-converting enzyme 2, or ACE2, in the brainstem, kidney, cecum, and hypothalamus. This, in turn, can lead to a dysfunction of the autonomic nervous system, meaning that the functioning of the heart, intestines, sweat glands, pupils, and blood vessels can all be affected. Finally, in some cases, exposures to environmental epigenetic factors can cause changes that persist for multiple generations. Through historical records, we can see evidence of transgenerational inheritance of epigenetic changes which may be transmitted through methylation of gametes. More about this can be found in one of your other epigenetics lectures.